those are all of our housekeeping items. I'm going to turn it back over to Will to introduce our faculty member.
That's great. Thank you, Maddie. And we're excited. We know that you're all here to hear this presentation we have and to get your questions answered from Professor Treadway, a professor here at Emory. Professor Treadway is a cognitive neuroscientist and licensed clinical psychologist. He received his PhD in clinical psychology from Vanderbilt University in 2012 and completed his clinical internship and postdoc at McLean Hospital, Harvard Medical School. Dr. Treadway is the director of the Translational Research and Affective Disorders Laboratory, or the TREAD Lab. The TREAD Lab seeks to understand the molecular and circuit level mechanisms for psychiatric symptoms related to mood, motivation, and anxiety. The lab employs a variety of methods, including a multimodal neuroimaging techniques like fMRI and MRS and PET, behavioral paradigms, computational modeling, experimental pharmacology, genetics, and clinical personality assessment. A particular interest has been the assessment of reward motivation in psychiatric populations, for which Dr. Treadway has developed several laboratory tasks, including the effort expenditure for rewards task. So if any of that is of interest to you, then you're in the right place. And with that, I'd like to turn it over for our presentation of the hour to the Winship Distinguished Research Professor here in the Department of Psychology, Dr. Michael Treadway. So Dr. Treadway, the floor is yours.
Wonderful. Thank you so much for being here. I've done a few of these over the years, and it's always really a pleasure to have a chance to interact with the future incoming class of Emory students. And I know it's a little different with the virtual format, but hopefully we can still have a good conversation. Just going to take a second here to share my screen and get started. Okay. As Will mentioned, I'm faculty in the Department of Psychology here, and I study both neuroscience and mental health. And the title of my talk today is Motivation and Its Discontents. Some of you in the audience may recognize that this is a twist on actually a seminal work by Sigmund Freud, one of the founders of psychotherapy, Civilization and Its Discontents. And we take that and we sort of focus a little more narrowly on a specific aspect of human behavior, namely motivation. And our research goals are twofold here. The first is we're interested in understanding how the brain makes decisions about when and how much effort to invest to accomplish anything. Knowing Emory students like I do and what it takes to get into Emory, the past 4 years, I'm sure all of you have become expert in this process of having times when you feel fatigued, feel tired, and yet somehow dig in to work a little harder, push a little harder, because you had a longer term goal of furthering your education and launching your career. And we're interested in how does the brain encode those kinds of abstract values, the value of higher education, for example, versus an option of just, binging on Netflix or hanging out or doing something. Not that there's anything wrong with those activities, but sometimes we need to push ourselves. And how does the brain make those kinds of decisions? What are the circuits, the brain areas, the neurotransmitters involved in making those kinds of decisions? So that's one of the things that we study as cognitive and computational neuroscientists. And one of the reasons we're interested in that, apart from the fact that it's just a fascinating question, one of the reasons we're interested is that those types of processes are really impacted in various forms of mental illness, chief among them major depression. And I'm going to be talking today a little bit about how we study the neuroscience of motivated behavior, and then how we use some of those tools and that understanding to develop new treatments for disorders like depression. And before I get into that, I want to spend a little time just orienting this group to why depression and other forms of mental illness are so challenging to study and so challenging to treat. And in many ways, it stems from the fact that when we talk about depression as a disease or addiction as a disease, we're using the language of a medical diagnosis, but it's actually really a very incomplete diagnosis. And let me give you an analogy. A diagnosis of depression is similar to going into a doctor's office and being given a diagnosis of fever. Yes, a fever is a measurable, recognizable, and reliable phenomenon that you can identify. But knowing that a patient has a fever tells you very little about what is causing that fever. What is the issue in the body that needs treatment in that moment? So it's very incomplete. And we know that fevers, for example, can be caused by a wide range of problems, everything from bacteria to an autoimmune condition to a cancer. And those different conditions require very different approaches to treatment. You're not going to get very far treating a cancer with antibiotics, and you certainly don't want to take someone who has strep throat and give them radiation therapy. But that is exactly what we often end up doing when it comes to mental health. In mental health, we have, mostly through trial and error and stumbling on things, we have developed a number of treatments that do work for some people. There are folks who get enormous benefit in their experience of depression from drugs like SSRIs or psychotherapy, or sometimes more invasive methods like ECT or deep brain stimulation. But for each of these techniques, there are many people who see no benefit. And moreover, our understanding of what is causing an individual's depression, where you sort of see all those question marks, our understanding is really limited right now. And so this is part of what makes studying and treating depression so difficult. Now, why do we not know what the causes are? Well, part of it has to do with the fact that there are many different potential causes to depression and causes that all operate on different timescales. Let me just walk through at a very high level some of the different causes to give you a feel. So first, there are what are sometimes referred to as early causal experiences, things that may happen early on in life that increase your risk for developing depression or other forms of mental illness, anxiety, substance use issues later on in life. And they include growing up with periods of significant trauma or significant life stress. And they also include genetic factors. Depression and many forms of mental illness, they do run in families, and they're often estimated in the case of depression to be about 40% heritable. That is to say, 40% of someone's risk for going on to develop depression is based on the genes that they inherited. But that's not the only way you can get depression. There's also adult causal factors, factors that emerge in adolescence and really any time in someone's adult life. Chief among them is chronic stress. What do I mean by chronic stress? Well, the last year comes to mind. For most of us, going through this pandemic has been a period of sort of unrelenting, low-level, sometimes high-level stress. As we've had to adapt to a number of disruptions and at times serious losses that we faced. That type of year-long period can lead to a real increase in depression. How does chronic stress and these early life factors and the genes you've inherited all lead to changes in the body and the brain? We still have a very incomplete picture here, but we know that there are changes in stress hormones, in inflammation that I'll be talking about, as well as changes in brain system functions that we can measure using various MRI-based techniques and other techniques where we see evidence of depression changing how the body and the brain function. We can also think about it at the psychological level. We can see changes and observe changes and patterns of thoughts and feelings that start to occur more frequently in the context of depression, more pessimism, less capacity to envision that things, even if they've been enjoyable in the past, that they'll be enjoyable in the future, and mechanisms of that nature. So these are some of the causes. And as I said, everyone's depression can be caused by a unique combination of these different factors, which is part of what makes it so hard to treat and so hard to study. When we study depression, we're often interested in trying to find something, some measure of brain activity, some measure of a hormone that might be useful in telling us this person is depressed and this person is not. To give you a concrete example of what we kind of hope for in this field is we want to see data that would look like this. This is made up data for illustrative purposes only, but we kind of wanted to find some measure where if you're below a certain score, like all these healthy control subjects, not depressed, you would be here. And if you're above a certain score, this red line, that would tell us you likely have depression. This type of test certainly exists for a wide variety of other conditions. In fact, diabetes works this way, where we use measures of blood glucose levels to determine if someone has diabetes or not. So you can have this kind of very clean result that's very useful in diagnosis and treatment. Unfortunately, when it comes to depression and most forms of mental illness, everywhere we look, we find a pattern that is more like this, where we do see a difference on average across individuals. So, study after study might show that on average, controls have different levels of stress hormones or different levels of brain activity in particular areas. And depressed patients on average might be higher. And that gives us some clue as to what might be going on. But the problem is it's not very useful at the level of individuals, right? Here you have an individual person who seems very high on this measure, but they're a healthy control. They're happy as can be. Meanwhile, you've got someone who's telling you they're really struggling with depressive thoughts, lack of motivation, other aspects of depression, and they seem very low on this measure. So we are struggling as a field when we just compare sort of groups of depressed patients to groups of folks without depression to find something that clearly leads to a cause and something that we can treat. So how do we go after this in our lab? We are focusing on one aspect of depression, which is really the reduction in motivation. We believe that if we can understand that circuitry, we can target at least that symptom, and we can identify patients for whom that symptom, that low motivation, may be a core source of their depression, and that might lead us to a treatment. It's not going to treat all forms of depression, but it may lead us to new treatments that some of our current treatments are missing. So I'm going to talk now just a little bit about how we study motivation and how we identify some of the neural mechanisms, brain areas, and neurotransmitters that support motivated behavior. A lot of this work actually began in a series of studies started in the early 1990s. Led by a very famous researcher in this field named John Salamone, who's actually an Emory alum. He did his PhD here back in the 80s. And John was really interested he studied rats. He did and still does study rats. And he was interested in trying to understand what made rats willing to work for rewards, what made rats motivated. So he developed this paradigm that you're seeing here. And what's depicted on the screen is a box. A rat is put in this box and is trained up to know in this box, it has a choice. It can either push a lever 5 times and receive a sweet pellet, Or it can ignore the lever, not work at all, and just eat this freely available but not particularly tasty lab chow. I actually, when I was a graduate student, I did a semester rotation in John's lab, and I can confirm the lab chow is not very tasty. So rats typically don't like this. They really prefer these sweet sugar pellets, and they will work hard to get these pellets. In a typical half-hour testing session, a rat will emit between 1,500 and 2,000 lever presses to get sweet pellets. And remember, the lever is about half the size of the rat. So that's kind of a whole body workout for these guys. That's a lot of lever pressing. And what's so interesting about this paradigm is John then used this to start disrupting different areas of the brain to see what disruptions would lead animals to not be willing to work for rewards. And he found two key brain areas. One is an area called the ventral striatum. So this is just a little neuroanatomy for those not initiated. An area called the ventral striatum, and in particular, a neurotransmitter in this area called dopamine. Now, you may have encountered dopamine in the popular press as being a neurotransmitter that is critical for pleasure or enjoyment. And that was actually a mistake that neuroscientists made in the 70s and 80s when they were first discovering dopamine. Really, what dopamine is about motivation. And when you block dopamine, you block an animal's willingness to work for rewards. And that's exactly what John found. He found that if he blocked dopamine, the animals would stop pressing the lever and they would start eating the lab chow. And what's so important about that is that blocking dopamine wasn't changing whether or not the animals were hungry. They ate the same amount. And if you gave them a choice between sweet pellets that they didn't have to work for, well, then they would eat those and they would prefer those to the lab chow. So blocking dopamine didn't change their basic hunger. It didn't change how much they preferred the sweet pellets over the lab chow. It just changed their willingness to put in work to get the thing that they liked more. And there was one other brain area that John and now many other researchers have found shows a similar effect if you disrupt its function. And that's a brain area called the dorsal anterior cingulate, shown here. And here's just some of what that data looks like. This is before and after a lesion to this area. That just means disrupting the function of the area. And you see that before, this is again in rats. Rats are choosing to work more between 80, 90% of the time. And then after the lesion, the animals that got a lesion, they're much less likely to work. And animals who got a sort of sham where they go through a surgery, but they don't actually get any lesion disruption, they stay the same. So that's fine for rats. What about humans? Well, I was inspired as a graduate student by some of John's work and wanted to see if some of these similar brain areas and neurotransmitters would function similarly in humans. And I'll just show a little bit of what we've done in this regard over the years. First, we had to develop some computer tasks to test human willingness to work for rewards, kind of like the rats in the box. And so we developed a variety of computer tasks like this one. You're seeing a schematic diagram of a single trial of one of our tasks, where basically on each trial, people are given a choice between doing a little bit of work for a small amount of money or doing a larger amount of work for a larger amount of money. And they make a series of choices in about 20 minutes. And we look to see how often do they choose to work more for more money. For those of you curious, the work here is pretty basic. It's button pressing. Psychologists love button pressing. I don't know what it is, but we do. And to make it effortful, everyone had to do rapid button pressing with their non-dominant pinky finger. So up to 100 presses in 21 seconds, which over the course of 20 minutes, that does get pretty effortful. And we found that if we gave folks drugs that increase dopamine, like amphetamine, we could increase subjects' willingness to work for rewards. Here you're seeing data from 2 studies showing that as you increase the dosage of amphetamine, you increase subjects' willingness to work for rewards. And that's now been replicated a few times. So dopamine seems to drive motivated behavior in a similar way in humans to what we see in animals. Now, that's a good start, but those tasks are a little artificial, a little limited. We've now, in more recent years, taken advantage of the wide availability of virtual reality platforms where we can look using tasks like this one. You're seeing a little video of someone going through a VR environment, and each room in this virtual world requires different amounts of physical effort to go through, and individuals have to maximize their points by collecting coins. And they, as part of this, they have to learn which coins and which rooms give them the greatest reward. There are lots of moment-by-moment decisions that go on in a simple task like this, where you're exploring a virtual reward, a virtual environment, and deciding, okay, these look like good coins. Do I stay here and collect them all? Do I keep exploring, looking to see if there's something even better? How does the effort it takes to move through impact that? Makes it sort of more real world decisions, even though this is still essentially a very simple, dumbed down version of Super Mario Brothers. And we can use tasks like this with techniques that can temporarily, very transiently disrupt neural function, just like the lesions to the dorsal anterior cingulate in rats. We can do something similar in humans using what's called TMS, or transcranial magnetic stimulation. That sounds like a lot, but it's pretty simple. You have this powerful magnet coil here that can deliver a brief magnetic pulse, and that will disrupt neurons in a brain area's ability to fire for just a second or two. And when you do that, you can then see the effect on behavior of disrupting a particular area. And it doesn't do any kind of long-term lasting harm whatsoever. I've had it done to myself many times, but it's a very powerful way of determining in humans the contribution of a brain area to a particular function. Here I'm just showing you the activation, identifying where we target for different people. This is their dorsal anterior cingulate. And here's just some initial data. We see that when we use TMS to inhibit the function of that same area I showed you in the rats, we see people's willingness to choose effort go down. Whereas if you target a different region that was included as a control region, we don't think it plays a role, you don't really see any effect. So that's just some of what we've done in terms of trying to understand the neurotransmitters and circuits that give rise to motivated behavior. And now I want to talk a little bit, transition a little bit to how we're using that information to understand what happens in depression, or at least depression for some people. So if we're correct, if motivated behavior is really dependent on intact dopamine and intact dorsal anterior cingulate function, this would mean that for some people, depression is maybe related to a reduction of dopamine. And that leads to the obvious question, what would cause someone's dopamine levels to go down? And one answer that we've been interested in that may surprise some of you is inflammation. Inflammation seems to be a potential culprit and a cause for low dopamine and depression. Let me walk you through some of what we and others in the field have been seeing. So first of all, what do I mean when I say inflammation? Inflammation just describes a sort of coordinated response of the innate immune system to damage, threat, and infection. And one of the key drivers of inflammation in daily life is actually stress. Psychological stress reliably results in increases in inflammation, and it also leads to increases in depression, as we talked about earlier. It's one of the main adult causal factors of depression. So stress can cause inflammation to go up and increase your risk for depression. Inflammation and being in an inflamed state is also associated with what researchers refer to as sickness behavior. Sickness behavior just describes how you act when you're sick. And if you think of the last time you had the flu, you probably felt really fatigued, didn't really want to get out of bed, felt kind of cranky, irritable. In other words, sickness behavior mimics some aspects of depression, particularly those related to fatigue and low motivation. And it's also been observed for several decades now that markers of inflammation that you can measure with standard blood tests are commonly elevated in depression. It's not in everyone with depression, but reliably 30 to 40% of patients with depression will have elevations in inflammatory markers. And one of the long-term debates in this field has been whether that's a cause or an effect. It could just be an effect. When people are depressed, they're tending very often to not be as active, to not be sleeping particularly well, to be experiencing significant life stress, to not necessarily have a great diet. All of those things can lead to inflammation. So many people have said, look, the increase in inflammation, that's just an effect of being depressed. It's not causing depression. But there's been sort of growing evidence that it actually may be a cause, at least for some people. And the strongest evidence for this comes from a series of studies that grew out of a kind of accidental discovery of a side effect from a treatment called interferon alpha. Interferon alpha is a drug that used to be used, no longer used, used to be used to treat various forms of cancer and hepatitis. And what it does is it causes a massive spike in activity of the innate immune system. You get really inflamed on this drug. And the idea is that this big recruitment of your innate immune system helps you fight off that cancer or hepatitis, and that can work. But there's a catch. And the catch is, for a subset of people, it makes them really depressed. And this is just an example of one such study. This was published in the New England Journal of Medicine, but there have been many others. This is a study done here at Emory by one of my collaborators. And what you're seeing is at the beginning of this study, 100% of study participants had no history of depression. They'd never been depressed. They didn't seem to have any risk factors of depression. And they were being given a life-saving, in some cases, drug, a treatment drug. So, they weren't depressed and they were getting a good treatment. What you see start to happen here is after up to 4 or 5 weeks on interferon alpha therapy, and a subset of them started to become significantly depressed. In some cases, the depression was so severe that the treatment had to be stopped because the risk of suicide was greater than the risk of cancer recurrence. So these are not casual sort of feeling tired, feeling fatigued type symptoms. This was full-blown depression. And it reliably occurs in about 20 to 50% of participants who receive this drug. So if you think back to what I was saying about all the complexity around what causes depression and depression for different people, here we have evidence that at least one reliable way to induce depression is by significantly engaging the immune system. So what is this drug doing? When you take interferon alpha and it causes this big immune response, what is happening? Well, follow-up study also done here at Emory wanted to ask the question, could it be doing something to dopamine? And indeed it is. When individuals receive this drug, they show a significant reduction in availability of dopamine. This is measured using a technique called positron emission tomography, or PET imaging. And we see a significant reduction in the availability of dopamine after 4 weeks of interferon alpha therapy. And this has actually been confirmed in animal models where you can measure dopamine more directly. Dopamine's a little tricky to measure using PET, but it is very clear when you administer a drug that causes inflammation to spike up, dopamine levels go down. And so how might that lead to depression? if you're all like many of the Emory students I've got to know over the years, I suspect you're really hardworking and you're really motivated to try lots of things and challenge yourselves. And that's also something that is a really important sense of your sense of self. It's part of what makes you feel really good as a person. So imagine what would happen if you entered a period where you had chronic elevations in cytokines leading to low dopamine, and this led to a spike in a symptom of fatigue shown here in red. What we see happens is over time, that chronic fatigue, you start to feel different about yourself as a person. You start to second guess whether you really are that motivated person. You start to get a little more pessimistic. Start to feel like things aren't really worth doing. In other words, some of those other symptoms that we think of as classically depression can grow out of this lack of dopamine-induced fatigue, even though the other symptoms aren't necessarily related to inflammation directly. So this is our model and how this line of research has grown out to explain at least a potential subset of cases of depression. So now I just want to talk briefly about what does this mean for treatment? Can we use this to treat depression? Well, if we're right that you have, potentially chronic stress and other factors leading to a spike of inflammation, leading to a reduction of dopamine, then there are two obvious things we can do. We can either try and block the inflammation or increase the dopamine. Both of those are being tried out with active clinical trials here at Emory, and we've already got some initial results. This was the first of its kind study that I'm showing you of a treatment for depression with a powerful anti-inflammatory. This is not like, Advil or Tylenol. This is a very powerful drug called infliximab. And what you're seeing here is patients in black who received infliximab and patients in white who received placebo. And the x-axis here is time, how long they were on the drug, and on the y-axis is improvement in depressive symptoms. And it's always plotted out, so lower is better. That means improvement. Depressive symptoms are going down. Now, at first glance, this looks like a really disappointing result, because while the folks in the infliximab group in black, they are getting better, they're not getting better in any way that is different from folks in the placebo group. But, and this gets back to what I was saying at the beginning, that different types of, that not all depression is the same. If you broke up the groups who received infliximab between the individuals who started out with high inflammation versus individuals who started out with no high inflammation, their depression didn't seem to be related to inflammation, then you saw a real effect of infliximab. It was only helping the patients with depression get better if they had high inflammation. So it seems somewhat specific to that subtype. And we think that that's a subtype, this what we call the inflammatory subtype, that is likely being missed by other kinds of treatments available. So that's one thing you can do. You can try and suppress the inflammation. And we have some follow-up studies using this drug and next generation drugs. The other thing you can do is you can try and restore dopamine. Do we have drugs to restore dopamine? Absolutely. In fact, that's the main therapeutic goal of conditions like Parkinson's disease, using treatments like L-Dopa to restore dopamine function. So one of my collaborators, again here at Emory, Jennifer Felger, has been doing that. And first, I'm just showing you some data that she collected in monkeys, showing that here, this is just what I told you before, that monkeys, healthy controls, this is a measure of dopamine. And then if you give them interferon alpha, that drugs that causes that massive spike in inflammation, dopamine levels go down. But if you give them interferon alpha and L-Dopa, shown over here on the right, you can restore dopamine levels. So maybe this can work in humans, and early results from one of her clinical trials using some of our motivation tasks show pretty encouraging results, where basically the people who receive L-DOPA are more willing to work for rewards on our motivation tasks. And as in the prior study, the effect is only true for people with evidence of high inflammation at baseline. So if you came into the study, you didn't have high inflammation, giving them L-DOPA doesn't seem to do much. Their depression may not be related to something with inflammation and dopamine function. But if you had high inflammation, giving L-DOPA improves motivation. This is a change score plotted out from baseline to end of the study. So I hope I've given you a brief overview of at least some of what we're doing in my lab and here at Emory to kind of use cognitive neuroscience to inform and identify new potential treatments for mental health disorders. And before we move to the question and answer period, I did just want to say a little bit about what brought me to Emory and why I'm so delighted to be at Emory. And there are three factors. The first is a culture of collaboration. I've been very fortunate in my career, my training, and where I've worked previously to have been at some really stellar institutions. Prior to coming to Emory, I spent time at Columbia, Vanderbilt, and Harvard, and they're all excellent places, but none of them have had, that I experienced, the culture of scientific collaboration and interdisciplinary collaboration that I've experienced since coming here. You will find that there's at pretty much every level, Emory faculty are on the lookout for ways that they can identify new areas of collaboration with people in other disciplines, other departments, and other institutions. With the basic philosophy that by bringing in groups of people who do different types of things and have different types of expertise, we'll do better science together. And that is not always the case. In other places, you may find that things are a little more siloed and there's a little bit more of kind of protective approach to turf and resources, and that's really just not been true here. And it's been, I think, a huge boon to me and helped me carry my research in directions I would never have anticipated. Emory also, even though it really is a college and has a college feel, there are top tier resources for all kinds of research here. I have never felt in any way limited to do any kind of technique I can think of since I've been here at Emory. If there's a new exciting technique published in the field, comes out in a paper of Science or Nature, it takes me about 5 minutes to locate someone here at Emory who knows about it, knows how it works. And if we decide it's the best way to answer a question, we're off working on it together. And that's really important for promoting cutting edge science. And finally, Emory really balances those, top two things I mentioned that really facilitate research with a commitment to undergraduate education. I've really enjoyed that here. I think Emory manages to have this very intensive, high-level research culture while also preserving the kind of intimate, small classroom feel of a small liberal arts college in many ways. And that's certainly been my experience and what I've heard from the undergraduate research assistants who work in my lab. Undergraduates are welcome usually by the time they're in their second semester, their first year, to join research labs and begin to get experience working on projects. We've had over the years in my lab, almost about 50 undergraduate RAs contribute, and they've been phenomenal. And they've helped with some of the data that I showed. They've had lots of creative ideas. Some go on to do honors research theses where they develop their own study and collect the data and analyze it. Some of them, end up doing some more on the clinical side, helping us recruit patients for our studies and get really experienced and comfortable learning how to conduct clinical interviews with folks struggling with depression and other issues. And so there's a nice wide variety of opportunities for undergrads to get involved. And they do great work. And so with that, I would just like to acknowledge actually the many people in my lab over the years, my collaborators at Emory, and just note that, I hope at least some of you will consider joining us if you come to Emory. So thank you all. And at this point, I'd be delighted to address any questions.
Thank you so much, Dr. Treadway, for sharing a little bit about your research and what happens here at Emory, and then wrapping it up there with a little bit about why you came to Emory and connecting, I think, especially for the admitted students watching this, what that means for someone who's about to enter their undergraduate experience who may be interested in neuroscience or psychology. We have a lot of students interested in those fields, and Maddie and I often get questions about it, but today we have an expert at Emory with us to talk about that and to answer some of those questions. And so we have some questions that were submitted at the point of registration. So Maddie and I will take turns sort of addressing those. And while we're doing that, we'd like to just remind anyone, if you have questions for Dr. Treadway about his research, about the academic experience at Emory, about neuroscience or psychology, please feel free to send that to us in the chat, and we'll keep, or in the Q&A, and we'll keep an eye out for those so that we can pose those questions to Dr. Treadway now as well. So I'll go to one of the questions that was asked, and maybe this is a transition question as we go from sort of research to students entering their first year. Someone asks, when can students get involved with research, and when do most students begin? Is it something that students can do in their freshman year? What would you have to share about that, Dr. Treadway?
Sure. I think, formally, there's really no restriction. You can start right away. And, sometimes we have students who start volunteering while they're still in high school. It depends a little bit on the specific lab that you want to work in and where they're at, and whether there may be specialized techniques or courses that you need to have happened to really understand what's going on in the lab. So that's kind of on a case-by-case basis. But I can say in general, I usually start getting requests from students usually after they've finished the first semester of the freshman year. They've kind of decided, I think I'm really interested in psychology. I want to learn more about this, and they start looking around for labs. And certainly by the second semester, the sophomore year, most folks who have decided they want to pursue a psychology major and want some experience in labs, they're getting matched up with labs. So it can start very early, although that's less common. But certainly early enough that you can get a solid 2 years of lab experience is typical for most of our students.
And I will just briefly add that I know that students coming in the freshman year or the sophomore year that may be interested in this, sometimes they get connected through professors that they have in the classroom, and often they connect through our undergraduate research programs office in the Office of Undergraduate Education. So there are a couple of ways for you who may be thinking about this. If you get to Emory, there are a couple of ways to sort of find out about opportunities available to you.
I want to ask a question that wasn't pre-submitted, but I've been thinking about it. So you mentioned that you came to Emory for 3 main reasons, one being collaboration, another being a commitment to undergraduate research and education. And I'm curious, how do you teach collaboration? How do you teach undergraduate students the importance of collaboration?
Yeah, this is a great question. And I think I think honestly, the best way is to teach by example. That's why those two really, to me, go hand in hand in such a powerful way. I think when, undergrads, when folks join a lab in a particular area, their first order of business, understandably, is to try and learn as much as they can about what that lab is doing. And that's correct. But they will then have the experience of that lab director bringing in someone from a totally different department for a collaboration they wouldn't necessarily have seen. And that offers the experiential learning of, oh my goodness, this is how we get to a project like we have. It came from, these two subfields that didn't necessarily initially look like they had much to do with each other. And so I think witnessing in the labs, and it's not just at the level of faculty, I should say, a lot of my coolest collaborations started with my graduate students. And that's often how it happens. It's sort of like parents often become friends through their little kids. Well, faculty often become collaborators through their students. Two students are hanging out talking about something and say, hey, this thing's like that thing. We should talk about doing something. And then the faculty meet and realize, yeah, wow, the students had a really good idea there. And so that is the best way, I think, to learn it, is to experience it and to see the power of collaboration. It's obviously something that's easy enough to say, hey, collaboration's good, but to really believe that, I think it helps to experience it. And folks here at Emory experience that a lot.
On the topic of collaboration and interdisciplinary study, we just have a question in the Q&A now from Kayla that asks, are students in your lab all either neuroscience or psychology majors, or is there an opportunity to do research across majors? And I think that I might expand that question out, not just to your lab, but just broadly at Emory. Is it most typical for students to join a lab doing research in the area that they're majoring in, or do you see some opportunities to sort of cross into different fields there?
Yeah, there are a lot of opportunities from different fields. It's going to vary a little bit lab by lab. My lab is, as you probably got a feel, really at this intersection of psychology, but also biology and neuroscience. And that's reflected in the majors in the lab. So I have a lot of psychology majors, a lot of biology majors, a lot of majors in a program called NBB, which is our sort of interdepartmental neuroscience major. Those are probably the main three. But, one of the things that's, I think, also indicative of Emory is you got a lot of people who are doing interesting combinations of things. I've had music majors in the lab. I've had anthropology majors. I've had sociology majors. So, and often they're double majors or a major minor, but there's certainly no limit in that regard. We just care that you are curious, that you're bright, that you're hardworking, and what you do in terms of your other coursework, that's for you. I myself was actually a music major in undergrad, and so I'm a big believer in the ability to blend aspects of the liberal arts and sciences.
So Sophia has submitted a question, and she said, are there any opportunities to receive credit for research work? And I'm going to expand it a little bit more, and not just as opportunities to receive credit, but when someone says, oh, I'm doing research this semester, what does that mean? Like, how many hours is that? Do you get paid? Is it a class? What does that mean?
Yeah. Great. So, it comes in a couple variety of forms. The simplest form is someone who's just interested in the lab and say, I want to get some research experience. I just want to volunteer. And you kind of, we like to have folks agree to a certain number of weekly hours for that, but it's kind of up to them. And that's about it. There are ways through, again, some of the different majors, each have their own way of giving course credit for working in a lab. And so it's usually around 3 hours per week over the course of the semester per 1 credit hour. And some majors require that a paper be written at the end, sort of talking about something you've been working on. Other majors do not. So that varies a little bit. And then what you're actually doing in those hours. So typically what students will do is they'll sign up for 3 to 4 hours. So it's 9 to 12 hours per week in the lab. And what you actually end up doing, that'll vary a lot lab by lab in terms of the project you're working on. But it's often the case that you're paired up with a either graduate student or postdoc or someone in the lab is working on a specific project, and you and that lab member divvy up tasks, things that need to be done, whether it's running participants. We have participants come into the lab and set them up with some of our computer tasks for them to do and fill out self-report questionnaires, and maybe go down to the MRI machine we have in the basement of our building to get scanned. We have undergrads who help do our neuroimaging. It may be that at that time, the data are collected, and what really needs to happen is to analyze some data. Often in psychology experiments, we're interested in looking at interactions between people. So common task may be to code up what's happening in a video of 2 people talking to each other, sort of coding it for different words or emotional expressions as part of a study. These types of tasks, or running statistical analyses on data that have been collected. So whole variety of different things depending on the lab and what the student is interested in.
We have a question from Abby who asks, as someone who wants to pursue clinical psychology, is there any advice that you have for an undergraduate psychology major? And I think that, that could be broad. I'll say briefly, every student who comes to Emory will have an academic advisor and a faculty advisor. So from an academic perspective, academic advising perspective, every student will have one of those available to them. But I think her question could be taken to, we're talking about research. Would you recommend any types of research or any types of experiences, maybe volunteering in certain settings, anything that you would recommend for someone interested in clinical psychology to perhaps get to know the field and if it's right for them?
Yeah, absolutely. And one of the things that I love about clinical psychology is there's an enormous amount of variety in what we do. So in my own career, I spend a lot of time doing research and teaching. I also see patients as a clinician, and those are all very different types of things, but I find each of them very rewarding. And what I would encourage someone thinking of pursuing this field is to really try and sample that as broadly as possible in your undergraduate time. You may come in thinking, I'm not at all interested in animal behavior or, studying how monkeys interact. But you may be surprised. It's actually pretty fascinating to learn about the complex societies that non-human primates create. Emory has one of the world's premier sites for non-human primate research. And so, when you see a course offered, and, I think we have one for undergrads, social behavior of primates, try it out, see what you think, because there are so many different ways to pursue a career in clinical psychology. Getting comfortable with the biological side of things, getting comfortable learning about some of the ways that people are thinking about psychology and artificial intelligence. All of these have implications for the future of clinical psychology. And so sampling that, and you may discover things that really move you that you didn't know about. I'll just say for myself, I mentioned earlier, I was a music major. I didn't take a single psychology class in undergrad. That was kind of a mistake in hindsight. I ended up going back years later to do sort of post-bac requirements. And at the time I was thinking I was just straight up going to be a full-time clinician. That's what I was going to do. I wasn't interested in doing research. And one of my first classes, showed me results from an fMRI study where they were looking at brain areas involved in how people manage their emotions. And I was just blown away. I had no idea this existed. I thought it was so cool, and it totally changed the trajectory of my career. So come in with an open mind, sample widely. That would be my advice.
Well, we've kind of reached the end of our time, and it felt like such a perfect ending. I don't want to, go any other direction, but thank you all so much for being here, for asking questions, for being attentive. I know I don't have a psychology background, so I find myself taking like lots of notes. I'm like, this is interesting. Like, I'm actually really enjoying this. So thank you, Dr. Treadway. We always appreciate you being here with the admissions office to share your expertise with these students who are figuring out where they're going to go next. And maybe that'll be Emory, maybe it'll be psychology, maybe they'll never take another psychology class and then end up being a clinician. You really don't know where life is going to take you. But thank you all for engaging with us today. And thank you again to Dr. Treadway.
My pleasure. Thank you so
Dr.
much.
Treadway. Congratulations, everyone.